In this video, we're going to look at gluconeogenesis. So gluconeogenesis is the production of glucose from pyruvate. But first we have to transport pyruvate out of the outer mitochondrial membrane and into the cytosol, which is where gluconeogenesis happens. Now, pyruvate can't just go straight back out through the outer mitochondrial membrane as it came in, so we have to go through this process in order to transport the pyruvate out, and I'm going to go through that now. So first of all, pyruvate can be converted to oxaloacetate through pyruvate carboxylase. And in this process, an ATP is used, and we have to add a carbon dioxide because this is a 3-carbon molecule and this is a 4-carbon molecule. So we have to add in carbon to get to the oxaloacetate. So the oxaloacetate can go two ways. It can be converted to malate, which can then be transported directly out of the outer mitochondrial membrane because there is a transporter that can transport it outside. Or it can be converted to aspartate, which can also be transported through the outer mitochondrial membrane through some transporters. So let's start off with the oxaloacetate being converted to malate. So it does this through malate dehydrogenase, and an NADH is converted to an NAD in this process. The malate can then be transported directly out, as I said earlier. So the malate then becomes malate dehydrogenase. Well, the malate gets converted to oxaloacetate through malate dehydrogenase rather, and an NAD becomes an NADH. To remember the direction, the rule about the longer name has the longer component applies. So NADH faces the oxaloacetate, and here NADH again faces the oxaloacetate. And that's a trick for me to remember what direction the NAD and NADH is going without needing to remember the complex chemistry that's going on. So as I said, oxaloacetate can either be converted to malate or we can get it to be converted to aspartate. So we can get it to be converted to aspartate through a transaminase, and there's more to this process. It's a transamination reaction, and we'll look at that more when we look at the urea cycle, but for now just accept that it's a transaminase. And then aspartate can be directly transported out, and then by another transaminase it can become oxaloacetate again. And just take note that I've written 2 beside each molecule. That's just to trigger your memory that, remember, at the end of glycolysis we produce 2 pyruvates. So in order to get back from pyruvate to glucose, we need to use 2 pyruvates and in turn 2 of every other molecule. Just keep that in mind when we're looking at this. So now we have this oxaloacetate, as I said, which is a 4-carbon molecule. We need to convert it to a 3-carbon molecule, PEP, which is part of glycolysis, as you would remember. And we can use PEP to go backwards in glycolysis and produce glucose in the end. So oxaloacetate is converted to PEP through PEP carboxykinase, which converts a GTP to a GDP. A GTP is very similar to an ATP, it just has a different name. So we'll keep that in mind, and because this is a 4-carbon molecule and this is a 3-carbon molecule, we have to lose a carbon dioxide in this process. So after this, we've got the same steps as glycolysis that are happening in between PEP to fructose 1,6-bisphosphate, except it's just backwards. So I'm not going to go through that because it is just the same thing backwards, and you can go have a look at the glycolysis video and draw out all of the steps backwards if that helps you, but I think it's just good to just remember that it's the same steps just backwards so you can really get to the nitty-gritty about gluconeogenesis. So the nitty-gritty is that fructose 1,6-bisphosphate gets converted to fructose 6-phosphate with a different enzyme, an enzyme that is not used in glycolysis. And in this process, a H2O becomes a phosphate. And the enzyme here that is different to glycolysis, so all of this has the same enzymes in these steps from PEP to fructose 1,6-bisphosphate. But here we change and we have fructose 1,6-bisphosphatase. This is pretty much the only regulated point of gluconeogenesis, so I'll just go through it now. AMP and fructose 2,6-bisphosphate will inhibit this because these promote glycolysis. On the other hand, citrate will promote this to go forward because citrate inhibits this in glycolysis. So it's just the opposite of glycolysis. And that's the best way to think about gluconeogenesis. It's the opposite except for the two enzymes, which the second of which I'm about to go through now. So we have fructose 6-phosphate to glucose 6-phosphate. And then we have glucose 6-phosphate to glucose, and this is where the different enzyme comes in. So this is the same, just backwards, but here we have a different enzyme that comes in. So again, the H2O is converted to a phosphate. However, glucose 6-phosphatase is the enzyme that is used here. And it is very, very low in skeletal muscle. It's pretty much absent. A lot of sources just say that it's absent. So that means that skeletal muscle metabolizes glucose 6-phosphate when it gets to this stage by glycolysis because it can't make new glucose. So that's just important to remember. Now, just another point. I'm missing an arrow here. So I just want to point out that the enzymes that are different compared to glycolysis are all of the irreversible steps. So this is step 1 and step 3. So these steps which are irreversible are different. And also the step from PEP to pyruvate in glycolysis is also irreversible. So that's also different. So all the irreversible steps are different. So that's why I added the reversible sign there. So you can clearly see that the irreversible steps are the ones that change in gluconeogenesis. Now let's quickly go through the number of ATP that we require for gluconeogenesis. So ultimately we require 6. So from 2 pyruvates to 2 oxaloacetates we use 2 ATPs because remember there's 2. We have to use 2 pyruvates to get to glucose because glucose ultimately produces 2 pyruvates in glycolysis, and since gluconeogenesis is glycolysis backwards, we have to make sure that we take into account that there's 2 of each molecule. Because pyruvate is 3 carbons and glucose is 6 carbons, so we need to get to 6 carbons in the end, so we need 2 pyruvates to do that. So then that's 2 ATPs in this step, and remember GTP and ATP are pretty much the same thing, they're just different names. And then we have 2 oxaloacetate to 2 PEP, and in this step we used a GTP, but remember there's 2 of each, so we multiply that by 2, so 2 GTP used there. And then if you look at the steps that I haven't written in here, there is a ATP that is produced from 3-phosphoglycerate to 1,3-bisphosphoglycerate, and that is just from glycolysis, so you can go check out the glycolysis video again to see how that backwards will produce an ATP, or will require an ATP. So see, this is where it gets a bit confusing. So in glycolysis we go 1,3-bisphosphoglycerate to 3-phosphoglycerate and we produce 2 ATPs, but since we're going backwards we're going to require 2 ATPs to go from 3-phosphoglycerate to 1,3-bisphosphoglycerate. So then you add those together, 2, 4, 6, and you get 6 ATP that are required for gluconeogenesis. And that is the basics of gluconeogenesis. And just before we finish, I thought it'd be a good time to just bring in the Cori cycle. So don't freak out, the Cori cycle is not a totally new cycle or a different thing. It just describes that from the muscle. So since, as we said here, glucose 6-phosphatase is not really found in skeletal muscle, that means that we can't create new glucose in skeletal muscle. So we have to transport lactate from glycolysis to the liver, which is shown here. If I just might just label that as liver so you can see it a bit more easily. So this is the liver. So we have to transport the lactate into the liver through the bloodstream, and then lactate can be converted to pyruvate. It's just the backwards reaction that we saw in the glycolysis video where we converted pyruvate to lactate. It's the same thing, just backwards. So we convert that to pyruvate, and then we go through this process to make glucose, and then the glucose can then be transferred back to the muscle to be metabolized by glycolysis, and it's just a cycle. So that's why I thought the Cori cycle would be good to bring in now. It's just an extra thing. It basically describes that while we can't make new glucose in skeletal muscle or in red blood cells, as I've written here, because red blood cells don't have mitochondria, we have to use anaerobic glycolysis to get to lactate, and the lactate can be transported to the liver, and then we can convert that lactate into glucose through gluconeogenesis and through the backwards reaction from lactate to pyruvate, and then pyruvate going through gluconeogenesis. So that just kind of describes how we can still work around the fact that we can't make new glucose in the muscle and red blood cells. So that was the basics of gluconeogenesis and a little bit on the Cori cycle.